My name is Dr Michelle Dickinson. I've learned that breaking things down is the easiest way to understand them. So join me. We'll break down scientific concepts, meet some amazing scientists and inventors, and help you discover the world around you.
Hi, I'm Dr Michelle Dickinson, and today we're going to be talking about genetics. What makes you actually you and different to every other living thing is your genes. Genetics is the study of genes and how they work. Your genes are your instructions for your individual characteristics, like the colour of your eyes or the colour of your hair. We inherit our genes from our parents, who inherited their genes from their parents, and so on. This can lead to family traits, like big ears or dimples when you smile. Genes are made up of DNA, which is a long molecule that contains the recipe to our own unique genetic code. DNA is crucial to life on Earth, and these instructions are crucial to how all living things develop, live, and reproduce. By the end of this episode, you will have learned about what genes are and how they relate to DNA. You'll also learn about how genes are responsible for what we look like and why we don't all look the same. We'll also do an experiment in which you will learn about population genetics and how to determine where you might have inherited your own family traits from. To do the experiments with me, you will just need your notebook and pen so you can write down your observations. If you don't have them ready, go grab them now. A little later in the show, I'll be doing an experiment with one of my science helpers who'll be joining us from home and speaking to one of my friends who'll be able to tell us even more about genetics and their research into diseases. Okay, let's get started. Your DNA is what makes you completely different from any other human alive today and any other human that has ever been on our planet. This is unless you're an identical twin, in which case you have the exact same DNA as your twin because you both came from the same initial cell and therefore were created from the same set of instructions. DNA stands for deoxyribonucleic acid and is a chemical found inside the cells in animals and plants. Mostly you find it in the cell nucleus, but some is also found in the power generator part of our cell called the mitochondria. DNA is made up of two long strands that are arranged in a spiral, a bit like a twisted ladder. We call this a double helix structure. The rungs of the ladder are made up of pairs of small molecules. These are called nucleotides, and there are 4 different types of these: adenine, cytosine, guanine, and thymine. We usually abbreviate them to their initials, so A, C, G, and T for short. We call these the base of your genetic code, and each rung contains a pair of bases. The order of these base pairs in the rungs of the ladder spells out the code that tells each cell how to grow, how to function, and how to reproduce. That is the genetic information that's in each cell. The sides of the ladder are made up of groups of sugars and phosphates. A gene is a section of the DNA ladder, which contains the information for one specific trait. DNA molecules can be very long. If you unwound the DNA helix in one human cell, it would stretch out to be 2 meters long. Most of us aren't even 2 meters tall, so this is very impressive. To fit inside a tiny cell, they are tightly wound together and scrunched up into little packages that we call chromosomes. Most humans have 23 pairs of chromosomes in each cell. Having more chromosomes doesn't mean being a more complex animal. While the fruit fly only has 4 pairs of chromosomes, the red king crabs have a whopping 104 pairs. You might be wondering why we talk about pairs of chromosomes rather than just telling you the total number of chromosomes that there are. And this is an excellent question. We usually talk about pairs of chromosomes because we get half of our chromosomes from each one of our parents. So in humans, that's 23 chromosomes from each parent. These chromosomes pair up with each other to make us. So chromosome 1 from your biological mother would pair up with chromosome 1 from your biological father, and so on. The fact that we get genetic material from both of our parents explains why we share characteristics with both of our parents. Let's use eye colour as an example. There's an area on one of our chromosomes that includes the genetic code or gene for eye colour. Let's imagine that the gene comes in two different versions, blue and brown. As you inherit one full set of chromosomes from each parent, you get two sets of the gene for eye colour. But most of us have either blue or brown eyes, and not one of each. If both of my parents have brown eyes, I'll have two copies of the brown eye gene. Easy, then I'll have brown eyes. But if one parent passes on the gene for brown eyes and the other passes on the gene for blue eyes, which one would win out? Well, my dad actually has blue eyes, but my mum has brown eyes, and my eyes are brown too. This is because the gene for brown eyes is what we call dominant, which means that if we have that particular gene, even if it's only from one parent, in my case, my mother, it will always win out over the other one. The blue eye gene is what we call recessive, which means that if we only have one copy, like I do from my dad, it's not as strong as the other one and so won't be expressed. Hence, my eyes are brown. If you inherit the genes for brown eyes from only one of your parents, you will have brown eyes. You need to inherit the blue eye gene from both of your parents for you to have blue eyes too. Confusingly, this doesn't mean that both of your parents need to have blue eyes. They might both have brown eyes, the dominant eye colour, but also they may have the recessive gene for blue eyes from their parents. Working out how different dominant and recessive genes are passed down from parents to children can be complicated, but it's important for people like doctors or forensic scientists to have this information. We're going to talk a bit more about dominant genes and the traits that you inherit from your parents when we do our experiment later on in the show. Forensic scientists are able to use the fact that everyone has different codes in their DNA to help solve crimes. As we've already discussed, DNA is found in cells, and wherever you go, you actually leave cells behind. These can be dead skin cells or strands of hair. Forensic scientists can collect these cells left at a crime scene and extract DNA from them. By analysing this DNA, they can create a profile of whoever was at the crime scene. The DNA profile is then checked against a database of other profiles. If there's a match, it could be used as evidence to place someone at the scene of a crime. Now that we've learned a bit about DNA and how it's inherited from your biological parents, let's do an experiment to see if we can figure out where some of our own traits have come from and how we compare with others in the population. Hey Hudson, how are you?
Good, how are you?
I'm good, thank you for joining me in a genetic test today. We're actually going to be experimenting on you, is that okay?
Yeah, that's fine.
Good. Okay, so we've been talking about genetics and traits that you can inherit from your family. And so do you have anybody who's a member of your family in the room today?
Yes, my brother and my dad.
We're going to need their help for this set of experiments too. So we're going to start with a really simple one. If you take a look at your ears or feel your ears, are the ends of your ears or your earlobes attached to the side of your head, or are they detached, meaning that they hang free?
Mine are attached.
Okay, now take a look at your brother. What are his attached or not attached?
Both of my brother and my dad are attached.
There you go. So that's where you got your attached earlobes from. So this is a very easy one that you can see in different people as to whether or not you've inherited it from your father's side, your mother's side, and whether your brothers and sisters also have inherited that gene. So you have attached earlobes. That's one thing. If you look at your friends, you will see that some of them have attached and some of them don't. And some are in the middle, meaning they're attached at the top or attached at the bottom. Now I'm going to ask you to fold your arms quickly like this. Okay, now take a look at the hand that is on the top. Is that your left or your right hand?
Left. For left, yeah. Left.
Okay, now get your family to do it very quickly too and see if it's the same.
Yeah. Yeah, both of them are left.
Okay, now try and do it the other way.
Oh. So left and the... Like that? No, that's it.
You'll find it's really hard, right?
Yeah.
Yeah, so you naturally fold your hand one way, and again, that's a genetic trait, and you'll find you typically hold it the same way that your family holds it. And it's almost impossible to try and fold it the other way. But for some people, it's totally natural for them to have their left hand instead of their right hand on the top.
Wow, that's awesome.
Okay, now there's something called a hitchhiker's thumb. Now, you may notice this in some people. I don't have it, but hitchhiker's thumb is where you can bend the top of your thumb backwards. Can you do that?
Yes, I can.
Can. Mine is totally straight. So take a look at your family. Can anybody else in your family do a hitchhiker's thumb where they bend this backwards?
Yes. My brother and
your
my
dad
dad
do
can
it?
do it. Yeah, both of them can.
Both of them can. So there you go. That is an inherited trait for your family that I sadly don't have. So hopefully I'm not going to be hitchhiking anytime soon. Now, those are all inherited traits, but there is something else going on alongside our genes that can shape the way we look, and that's our environment. Let's take the example of height. Genetically, you may be predetermined to be a certain height when you finish growing. But the height you end up at can also be determined by what happens in your childhood. Now, I'm hoping that you're having a happy and healthy childhood, but in times of famine or times of high stress and malnutrition, that can affect how much energy your body has to grow. So you'll never actually reach the full height you're predisposed to be. This is a really hard one to test. So when you're a grown-up, let's say your dad is a certain height, we don't know whether his height today comes from his genetics, his environment as a child, or some interaction between the two. We call that interaction between your genetics and your environment your phenotype. And this is where our genetics is only one part of how we look in the end. Okay, before we go, Hudson, I'm going to do one more test on you, and it's tongue rolling. Can you roll your tongue so it curls over from the sides like this?
Yes, I can.
Okay, what about your family members? Can your brother do it?
Yes.
And what about your dad?
No, he can't.
He can't do it. So do you know if your mom can roll her tongue?
Yes, I think she can.
So there you go. That shows that your mum's genes have passed down to both you and your brother to give you this amazing tongue rolling skill. So Hudson, we can keep testing on our friends, on our family, and work out what our dominant genes are and where they've come from. Was that interesting?
Yeah, that sounds really interesting.
Awesome. Well, thank you so much for helping me today. I'll see you soon.
See you soon. Bye.
Okay, after any experiment, we need to write down our observations. These help us to understand our results and make interesting predictions for our next experiments. So, our observations. What did we see? We saw that there are traits that we both have, like attached earlobes. Hudson, his brother, and his dad all have attached earlobes, and they all fold their arms in the same way. Hudson has hitchhiker's thumbs, and again, that trait is shared by his dad and his brother. Hudson and I can both roll our tongue, and Hudson inherited this from his mum. There are also some traits, like our height, which are hard to pin down to genetics because they depend on our environment as well as on our genes. Now let's look at our observations and reflect on them. By asking questions about why something happened or how we might make it happen again, we're really starting to understand the science behind it. We've only done this experiment with 2 people, and that's myself and Hudson. That's far too small to make any conclusions about traits in the general population. For this to be more scientific, we need a much larger sample. Also, as we know that we inherit our traits from our parents, we would need to know whether people that we're looking at are actually related. You could learn about inheritance by talking to your brothers and your sisters, and then widening it to your parents, your aunts, your uncles, your cousins, or even your grandparents. Why not compare traits with your friends to see how many you share and how many you don't share? A simple one to look at is your earlobes. Are they detached or attached? What about your friends? Okay, now we have a little bit more understanding about how our genes are passed down through families. We're going to talk to somebody who studies the link between genes and diseases. Dr Melanie McConnell is a geneticist at Victoria University of Wellington. Her research helps to develop more effective cancer therapies by looking at cellular survival pathways in the hope to find better treatments. Melanie, how are you?
Hi, I'm very well, thank you.
So, before you became an official scientist, can you talk about your childhood? Did you love science as a child?
Science is always, I think I've always had a scientific perspective on things. I didn't always love science at school. And I think there's a difference in thinking about a problem in a scientific way and then the kind of stuff that sometimes they get you to do at school, which they label science. But I think I was always quite into questioning how things worked. And to me, that's really what science is.
What is it that you do in your job?
So in my job, I get to ask questions about cancer cells. And what I try to do, me and the team of people that work with me, we try to understand how cancer cells form and how they live and how we can kill them. So that's really what we're trying to figure out is what is it? Can we tell when you look at a cancer, at a tumour, at a growth in somebody, can you tell what therapy you should use? Can you tell if that therapy is going to work? Can you tell what can you predict about a tumour by looking at its DNA sequence particularly?
And so is there something special about cancer cells that mean that they grow differently or they behave differently to our normal cells?
So normal cells, there's a set of rules that cells live by, and that set of rules is written in the genome, so it's written in the DNA, and in that DNA is written all the rules that a cell needs to live by and to behave nicely and to play with other cells and to go from being a fertilized egg and go through all those cell divisions to turn into a person. And the rules are quite complex because we're really complex organisms. And what happens in cancer is something goes wrong with the instructions. Something goes wrong somewhere in our DNA. It goes wrong with the instructions and a mistake happens. And a cell loses the ability to follow the instructions anymore. And if you don't follow the instructions, things can go very badly wrong.
And so let's talk a little bit about how you do this. You have a lab, I'm assuming. So what's in your lab and how do you manipulate or even look at these cancer cells?
I guess the main tool that we use in my lab is what we would call an incubator. We get cancer samples, and what we do with these tumours is we chop them up, dice them really finely, and then we put them into these plastic dishes. We keep these dishes in these big incubators, and so inside it, the incubators are sort of, this big, and they are very warm. They're really humid. They've got lots of, the environment mimics as much as possible we can inside a body, and we can grow these cancer cells in these dishes. And so we'll use light microscopes to look at them. We can grow them, and we can maintain these cells for years and years and years. Because the cancer cells are so abnormal, they will just keep on growing. And so we just keep these cells growing, and we grow them in the lab, and we can try to figure out what's making them tick.
It sounds like you're doing really interesting research, and there's so many things that you could be looking at. What is it that you enjoy most about your job specifically?
I guess I love that I've managed to work quite hard and get to this position where I can ask questions for a living. I come up with these questions about why does this cancer cell not die when it was treated? And then we get to figure out how to answer them. The best part of that would probably be figuring out that your answer was wrong, but that's because your question wasn't quite right, and then working out what it is you actually need to do.
If I were to ask you to sum up your research in one sentence, what would you say?
Cancer cells are smart. They're really hard to kill, but if we can be smarter, we can work out how to get them.
Melanie, that has been fascinating. Thank you so much for your time today. And I think you're probably going to inspire a whole bunch of new cancer researchers along the way.
That's been a pleasure, Michelle. Thank you.
There is so much still to learn about DNA. One thing that has always fascinated me is the science of cloning. I often say, I wish I could clone myself so I could get more things done. but I'm not actually sure I would want a clone of me walking around. Cloning is where we make a complete genetic replica of an individual from their DNA. Humans have been cloning plants for hundreds of years just by taking a small cutting from one plant and regrowing it from that piece. Around 20 years ago, scientists thought that cloning animals and humans could be the future of medicine and put lots of research time into the most famous clone of all, Dolly the sheep. While Dolly started her cloned life in a test tube, there are actually animals that naturally clone themselves. Hammerhead sharks, fish, jellyfish, lizards, and some snakes can all produce clones of themselves. Jellyfish are really interesting, as they can make clones of themselves while they are still growing. A young jellyfish is called a polyp. The polyp clones its own DNA and puts those clone cells into a little bud, which detaches from the side of the polyp and swims away. Some of these clones continue growing into adulthood, meaning the DNA from one jellyfish can be found in hundreds of different individual jellyfish. I guess it's a good thing that jellyfish don't commit crimes. Komodo dragons are some of the biggest lizards on Earth, and the females can produce eggs with cloned offspring when there are no males around. Actually, isn't that the story of how Jurassic Park started? Well, we haven't managed to clone a T-Rex yet. A few years ago, biologists managed to bring parts of cells from the extinct woolly mammoth back to life. DNA was extracted from the cells of a dead mammoth that was around 28,000 years ago and inserted them into cells from a modern-day elephant. To everyone's surprise, the mammoth DNA started to wake up and activate. Paleogenomics is the field that looks at the DNA of extinct species, and this can help us to understand the process of evolution. But we're still a long way from bringing a mammoth back to life. Some scientists are trying to create a mammoth-elephant hybrid, which I guess you would call a mammophant. And this is where the ethics of what we should and shouldn't do around science start to come to play. Every living cell contains DNA. Our genetics make up who we are and what we look like, and we can thank our biological parents for these traits. Genetics make us different, while also keeping some things the same. New research is being done around genetics all the time, and there's so much more to discover. Some genetic research is helping us to modify plants to help feed the population by being more resistant to changes in the climate, while other research can help us to understand how to diagnose, prevent, and treat diseases in people to help prolong life and reduce symptoms. DNA analysis can also help us to solve crimes, even decades after they have happened. While the study of genetics is fascinating and potentially life-changing, it's also a topic of ethical debate as we make decisions around what we should and shouldn't tinker with in this research field. Teachers and parents, don't forget to visit nanogirlslab.com, where we have downloadable content to help you to continue on your science journey into the amazing world of genetics. Thanks for watching. Do our genes decide how long a person will live? What's the most common hair color? Is height genetic? How do DNA mutations happen? How is plant and animal DNA different? Why is genetic variation a good thing?